Hi everyone. We'll wait a couple of more minutes to let time people to arrive. It's my great pleasure to talk to you, and I would like to really thank all the organizing team, because it's really amazing to be able to speak to you from France. So okay, we'll start. So hi everyone, it's my great pleasure to talk to you today mainly about our MS Bioscreen project and how it started. And it's truly a work that we've been experimenting, going from bedside to bench and eventually back. It all started with MS genetics, which is something that our group based in San Francisco have been really involved with. I'm myself a faculty from the Department of Neurology at University of California, San Francisco. And I want to disclose that I've been the founder of metadonics.com, it's a company doing biostatistics and as a service in 2008. And I'm the founder of Mira Medicine in the USA last year, which is offering personalized medicine development. So let's go. Just going to show you the slides, and we'll talk about this MS Bioscreen project, which essentially is an iPad application. So usually the best that I do is actually doing a live demo. That's really the best way to see that this is a prototype for real. So instead of that, what we'll do today is really kind of a video that we prepared showing what it is about. The Bioscreen is really a project from an entire team of people that you can see here with the background our great building in the Mission Bay campus, south of San Francisco, with view on the Bay Area. It has really been possible by a team of people that have been really helping so a core team of people that really have been working with us on the more technical part of the project, but also a great team, extremely. Okay, sorry. So here, what I wanted to show. Great team of people, the core MS Bioscreen team, with a lot of skills in terms of data organization, data analysis, and clinical data, but also the contribution of a dozen of key faculty from the UCSF MS Center and investigator of MS EPIC cohort. So let me just tell you a little bit about MS, since it's all about MS in this session, and maybe also the view we have on MS, affecting about 500,000 people in North America, 2.1 to 2.3 million people worldwide, and truly the leading cause of disability among young adults. And really, what I think is very important is what it means to a patient. The initial symptoms are there at age 20 to 14. It's a lifelong disease that you have to live with for a very long period of time, but with perspectives of progressing disability. There's no cure, there's disease-modifying treatments, but these treatments are very expensive. And more importantly, and I think we don't really realize what it means to patients, but patients are totally in the dark about the progression. And if you think about it, it's extremely difficult to have a disease where you don't really understand why you have MS, even if we know that genetic factors are very important. This is very important to remember that it's not like an infectious disease where you're going to say, well, at that time I was infected by a virus or something. And more importantly, once you know you have MS, you know nothing about the future. You don't really know about how it's going to progress. And truly, there's a very, very large variability from one patient to the next. So MS is very interesting because it's really a challenging disease with very complex clinical decision making. Evaluating MS is very complex because you have to integrate a lot of different variables and factors. It's truly a multifactorial disease with the interplay of genetics and environment. There's not only immunological manifestation, and as I said earlier, a lot of variability over time for a given patient, and also across the patient over time. So one difficulty that is more and more important in MS is really choosing the right treatment. So many, a few years ago, there was not so much too many treatments to think about. And now we really have many different therapeutic options. Each of them has a different efficacy profile and also a different risk profile. And it's not that easy to say what is the recommended guidance and protocol. So what's happened is really kind of a trial and error process that is pretty common in MS. To try to find the safest drug with the maximum efficacy for a given patient. So the question really we had with this project was really thinking about how can we think about integrating all the information we have, all these different factors that are important for MS, whether they are coming from MRIs, the results of clinical trial, genetics, etc., etc. And that's how we start thinking about the MS Bioscreen project as an integrative platform that will be able to take into account all the information. But before I'm showing that to you, I want to step back and tell you a little bit about the origin of this project, because our group has been very strong in genetics, and we've been, with many other groups, in the world, we've been founding the International MS Genetic Consortium, who frankly has been very, very successful in advancing the understanding we have of MS and MS susceptibility from the genetic point of view. So I wanted to give you an overview of a very summary overview of what can we say about MS, and the best to do that is really to have an historical perspective. So I like to present the genetic of MS by looking at really the one result we had for about more than 30 years now. It's really the finding that there's some genetic polymorphisms in the MHC region that are associated with MS susceptibility. And for 30 years, that was the only genetic marker that was known to be associated with MS risk. Many others were tested. And until 2007, when there was the very beginning of the genome-wide association era, and by looking at using molecular genotyping experiment and automation, we were able to finally discover new new genetic marker, and including the IL-7 receptor polymorphism that has been, since 2007, one of the most important gene associated with MS beside HLA-DNA genes. As you can hear, I'm nearby the hospital here in Nantes, in the west of France, and I apologize for hearing all that noise in the background. So since 2007, this GWAS era really changed our understanding of MS. And in particular, outside the MHC, we found many additional genes, so namely 16 in 2010, 62 in 2012, and a few months ago we published another Nature Genetic paper where the number of loci that are associated with MS risk is about 110. So if that looks extremely successful, because we went from 2 regions in 2007 to 110 in 2014. The other part that is very important is really to look at not only the number of genes, but the complexity of the models we need to think about genetic associations. And that's typically the case for the HLA. That's true that we had only one region for a very long period of time. But the model that explained the MS risk actually became extremely complex over the years. And they are extremely difficult to understand now, because we need haplotypes and diplotypes, which are basically pairs of haplotypes to properly describe the genetic risk in that particular region. The reason I'm saying that is that the genetic was a typical problem in MS to say, hey, we went from one gene that was well known and easy to remember, easy to understand, to a gene which require more and more complexity to detail and to understand the type of association with multiple sclerosis. And the same is actually currently happening outside the MHC, when more than one genetic marker is needed to describe the risk in a particular region. So it's really a quantitative change, the number of markers, and it's also a qualitative change because the complexity of the models have been evolving. Overall, and it's very interesting to remember in our own paper, we show that the variance that is explained by the MHC is actually just about 1/3 to a half of what we're able to explain using the rest of the genome. So more than 100 markers on one hand, one region on the other, the MHC, that still explain a lot of the risk. The reason why I wanted to show you that before talking about the bioscreen is because back in 2010, 2011 in our group, we say, hey, something is very much changing. When you look at what happened here around 2011, 2010, we start to have more genetic markers that we can remember. This is really an illustration of how complex this neuroscience, neurogenetics, and biomedicine in general is becoming. Because we have the GWAS and all these associations, we were able to discover a great deal of knowledge in genetics. If you think about it, what we're doing is, in this genome-wide association analysis, we're taking thousands of patients, we're taking thousands of controls, we're taking hundreds of thousands of very simple markers, the single nucleotide polymorphism, and we're comparing the cases to the control and using a bunch of statistical methods to make sure that we truly have a little bit of excess of one particular marker in cases versus in control, in MS, in people with MS compared to people without MS. And that was very successful because we now have 110 genes that we know for sure have something related to MS risk. But that's create really a problem because even if you're a great geneticist and very much involved, you can't remember all these genes. And it's also a little bit frustrating because all this GWAS is really a matter of comparing thousands of cases and thousands of patients and thousands of controls, and the next step is just to get bigger and we'll find more genes. Great. But that's also created a new opportunity to actually say, hey, instead of looking at thousands of patients and thousands of controls for one marker, maybe we can look for a great number of markers in one individual and see how much of these markers of these risk alleles are actually accumulated in one single individual. And that's where we started to design methods with many others called genetic risk scores, genetic risk profile, where unlike in the GWAS where you are at the population level, you can go back to the individual, go back to the bedside, and start to have a profile of an individual patient. And you'll take HLA DRB1 and see if the risk allele is present. You'll take the next one, IL-7 receptor, discovered in 2007, and you'll see if the marker is present. And you can take the other ones, and etc., etc. And now we're doing that for 110 genes. So we're now able to go back from the research population-based analysis, the GWAS comparing thousands of patients, to an individual level of analysis where we can summarize in a mathematical score, which we call the multiple sclerosis genetic burden. We can summarize how much one particular patient accumulates lots of genetic markers. So that's really important because, again, what we need is really to catch up in a way with all the literature that studies genetics in multiple sclerosis. And again, I'm taking that as an example of how much the knowledge is growing and how much we need to have access to information at the individual level. So we usually describe the knowledge about MS genetics into versioning, using a versioning system, pre-2007 on the HLA. And with the first GWAS, we had like a version 1 around 2010 with 16 SNPs, a version 2 of the knowledge in MS genetics with 62 SNPs, and a version 3 with 110, which is almost recent one, but sure, a version 4 is also coming, and we are really working on that. We summarize and explain that in a review in immunological reviews, and we also show that these markers are extremely diverse in terms of frequency and also their odds ratio, so namely the effect size and their contribution to MS susceptibility. So the evolution of that risk really also shows that our knowledge is increasing over time. But if you think that the knowledge is increasing kind of quantitatively, because usually people would say, hey, this is great. You had 17, then you had 62, and then you had 110. You guys are fantastic. The knowledge is really growing from one version to the next. Actually, this is not exactly true, and I'll show you why in that slide. I cannot show you this web link, but this is essentially a screen capture where what we show is essentially the markers, their risk and frequency, and the effect size. And we'll show larger circle here if the effect size is greater. So we're gaining more statistical significance here, so we're going up on average. We're obviously getting more, but what we can't show in here is actually even the description of a particular region is changing. So it's not necessarily sure that the genetic marker we use for one region in 2010 will be the same in 2011, 2012, or in 2013. So for genome-wide association studies, it's not so much of a problem because we're usually obsessed by p-value, that's the statistical significance of the finding, we certainly don't want to report a false positive. So we're very, very cautious about publishing regions that we think are truly associated with MS. What marker we're going to use, we don't care so much, and we don't look so much at their properties. But if you start to use these markers to describe an individual risk, you're doing something very different. You're not trying to find a statistical correlation between MS and the presence of a particular illness. What you're trying to do is you're trying to categorize patients. So if you use one SNP in 2010 with a frequency of 10%, well, 10% on average, 10% of your patients will be, and maybe a little bit more, will be categorized as at risk for that SNP. But if you're changing SNPs in 2012 and you're actually using a little bit more frequent marker, because a more frequent marker gave you more statistical power, and since you select that by p-value, what's going to happen is you're going to re-qualify patients that were not at risk for the same locus in 2010. Now they're going to become at risk for 2011. So what it shows and what it suggests is that the linearity of the number of SNPs and regions that we know that are associated with MS and their quantitative increase does not at all reflect the complexity of the association and the fact that we change gear and may use a different SNP for the same region. Again, it's not a concern for GWAS because they're looking for great p-values and statistical significance, but it's very important for when you try actually to categorize patients and find an individual way to summarize the aggregation of genetic risk allele at an individual level. So I'm not going to talk more about this MSGB and genetics. There's plenty of papers that have been published in Annals of Neurology in 2007. We published 3 last year looking at the properties of these risk scores and how important it is to be able to summarize the individual genetic burden. So you can look at these papers. In this talk, I wanted to talk about genetics for a little bit, just to illustrate a trend that is absolutely true in multiple sclerosis, but it's probably true in other type of data for multiple sclerosis. We used to be able to handle a limited amount of information, one HLA gene, a few dozen of SNP markers that describe the risk of MS. It's not possible anymore because we have 100 clones. It's not possible by the quantity of SNPs. But even more importantly, it's actually not possible anymore because of the complexity of the association. Even for a particular region, if we're changing the marker that describe at best the association with MS, if we're changing the allele, if they have different frequency, that is becoming a nightmare. And if I go a couple of slides back, right here, it's actually exactly what happened for HLA. We used to call the HLA allele HLA-AW2 because of the serology that was used at the time, and then it became DR2 because new antibodies were available, and it became HLA-DR15 with serological techniques, and then HLA typing evolved with molecular techniques, and it became HLA-DR115 where the polymorphism were looked at by PCR, and now we're sequencing all the exons of the genes, and we're really refining the association. So think about that. And it's true for genetics, and it's true for many other fields. A lot of new data, and an exponentially growing complexity of the associations. Very true for HLA with 20 years of history, and very true for the SNP outside the MHC, and now we're talking about 100 regions. So that kind of creates the need for Bioscreen, and what I'm showing here is actually a little preview of the genetic area, the biomarker area of our Bioscreen application, where we are making available the information about genetics at the bench. So we have patients that have been typed for all the genes. This is version from 2012, so we have 62 markers. All the patients are typed for these markers, and we provide a graphical summary of what genes are actually available. That's on the right panel in here. So this is typically a patient that is positive for HLA-DRB1, 1501, and that represents a certain amount of the known genetic risk, 12.5%. So we show a slice in the full pie of known genetic risk that display HLA, and all the other small slices here are actually other risk allele from markers at different loci that are also present in this particular patient. So if you want to summarize how much this patient has in terms of genetics, where we can compute a score that represents a weighted sum of the log odds ratios for each of the risk allele. It's very difficult to interpret, and I actually don't think we should try to interpret it too much. What we can do is actually interpret this kind of information by comparison to a reference group, which are here healthy controls. So this 10.4 number that we have, this genetic risk score, this MSGB, MS genetic burden, that we have for this one patient, is really a score that is difficult to interpret. But what we're doing here is we're saying, hey, compared to about 500 healthy controls, what we can tell you is that only 2% of the healthy controls have such a high genetic risk score. And so we're making a personal interpretation of the genetic risk score by comparison to a reference group. And that's pretty much how the MS Bioscreen started. Say, hey, if we're doing that for genetics, if there's a need to try to translate that and get back from the genetic research project to an individual vision of what are the genes that are, what are the risk alleles that are present, how much this patient is loaded. So that's probably true for other type of variable as well. And typically in this example, we have a suspicion that these patients might be pretty genetically loaded compared to other patients in healthy controls. So we realized, say, what we're doing in genetics is really having a lot of data that we've been crunching to really understand MS genetics, but that's actually true for many other dimensions of the data. And we're very lucky to have this MS EPIC cohort, which really represents 8 to 10 years of follow-up for about 600 patients. So it's about between 4,000 and 5,000 patients a year. So this is a lot of data. It contains clinical information, MRIs, treatment, genetic, and different type of genetic. And we want to address the idea, the problem that this data is not really used by so many clinicians, and it's not even really easy to use for researchers and many other people. So we saw that maybe this data set should be better accessible at the individual level. And let me show you, to be better accessible to really address this idea that there's many therapeutic options, and we want to accelerate the path to finding the right treatment for the right patient. So again, in MS, the challenges are evaluate the disease, how severe the disease is, how fast the patient is progressing. Challenge number 2 is to select the right treatment at the right time. And because it's so complex, there's really an opportunity to educate and better communicate with the patient using reference data. And maybe it's also the same case to some extent for both clinicians and researchers. We need to better understand the data that we have. So what we really try to do is say, okay, let's take an individual patient. Let's force ourselves not to work with thousands of patients as statisticians or epidemiologists. Let's work like clinicians with one patient. But we want to integrate the information about clinical data, motor function, genetic information, brain and eye imaging, and eventually spinal cord, having all the information about treatment, environmental exposure. And what we want to do is answer for the patient how her particular multiple sclerosis is evolving, how this one patient is progressing. So then we'll probably show the video. We try to show you a couple of examples of how we're showing these screens and how we're trying to do that using this iPad application that we have for multiple sclerosis. And if I switch here for a minute while you're starting the video. So this is really an iPad application that I have here on my iPad mini. It's connected directly to the data, to the database we have in California. It's a secure access, and you really have all this information. So I'm not sure. Are we done? Are we starting the video now? Should I do it by desktop sharing? Okay, no, okay, excellent.
The MS Bioscreen is the first precision medicine tool developed here at UCSF by a leading team of scientists and researchers. It is designed to bring the latest research in multiple sclerosis to the bedside. Take Anne, for example. She is 31 years old and was diagnosed with MS 10 years ago and has been on aggressive therapy for almost 2 years. She has been using a cane for walking and has had trouble with spasticity and balance. What Anne wants to know is how her disease is likely to progress, how she compares to similar patients, and what course of action will best treat her symptoms and slow down the disease. The MS Bioscreen brings a new look into the disease in 3 ways. First, during consultation, Dr. Smith can access all of Anne's data and instantly get a snapshot of how her disease has been progressing over time. Second, Dr. Smith can compare Anne to similar patients to provide a frame of reference that is tailored to Anne's characteristics. Dr. Smith can also very simply access other types of data from this tool, such as imaging. Thirdly, the enhanced comparison enables Dr. Smith to assess the probable path of Anne's disease, which informs a more precise and personalized therapeutic decision-making. The MS Bioscreen is also a great way for Dr. Smith to share information with Anne so she can be equally informed of her disease progression and actively participate in her treatment decision. This is a new way to practice medicine that is evidence-based, data-driven, and highly personalized to accelerate the path to the right treatment for each patient.
Thank you very much. So again, this is really a prototype that we've been developing at UCSF. It's a functional prototype that we have available for all our 600 research patients, and the ambition is really to be able to access right here from the iPad the data of any of our patients. So you can see here, basically connecting to the central database, getting access to the treatment information, to the clinical data, to metrics that we compute from MRIs, and genetic information. So using the slides that I have in here, and the video is pretty quick, I want to go back on the main features of that tool. The first thing is we aggregate all type of patient data into a single view, and that's typically the home page that we have in here, and basically we have all the basic information about the patient, and including the history of what happened in the recent years and what treatment were used, when there were relapses, and etc. That's kind of a patient overview and kind of a homepage. We also want to give access to information that is not typically easy to access to. And a very good example of that, switch back, is 3D, 4D MRI data. It's also true for genetic and biomarkers. They're not usually available to clinicians and to patients. But typically, we want to solve this data accessibility problem. So we have images for 6 years in a row for patients that had MRIs. We want to access the information. So we developed a proprietary system where we have the MRI stored in the cloud, and through the Wi-Fi, we pull them down to the iPad, and we can look at them not only 3D, where you can actually move the slice and the cuts in the brain space, but also over time, where you want to actually see how a lesion is progressing for one particular patient. And more importantly, we implemented, since the data is available for one patient, we implemented comparisons that are tailored to the one patient we're looking characteristics. So that sounds complicated, but the best way to understand what we're doing is to think about pediatricians. What pediatricians are doing when they have kids, they weigh the kids and they measure them, and essentially what they're doing is they take your baby boy or your baby girl, they weigh them, and instead of telling you, hey doctor, I think your daughter she's a little petite, and kind of having a subjective assessment, well, what are they doing? They're actually using growth curve and say, hey, if I take the weight of your daughter in that particular growth chart that represents young babies in America, I can tell you that your daughter is in the bottom 2%, so you really have to feed her. So that's really what we call data-driven evidence. That's really what we wanted to do with the Bioscreen by unleashing access to any kind of data. So for that, we developed really a two-hand infrastructure, a back-end infrastructure that contains all the information available from the cloud, and let's make that available to the application. And the application really represents the front end of this knowledge and data infrastructure, where we present all the data. You have this overview. Again, we're showing for one particular patient what is the data that is available. And it's also secure, and we really developed a HIPAA compliant infrastructure. So all the names that you're seeing now are actually fake. We use mockups for all the PHI information, but security is taken very seriously. And when you look at a particular metric, and this is typically EDSS, one of the most used clinical functional disability scale for multiple sclerosis, you can actually see in blue, number one, the individual trajectory of a patient. So what over time, what is trajectory over time? Two, you can, and that's the bottom bar, you can find and the application proposed to the user using an algorithm, a reference population to be used to compare that patient. So basically, patient with the same disease duration. And from this reference set of patients, we're computing the orange background, which provides a normality assessment, and that's the search feature that I wanted to stress on that screen. And the solid orange line represents the median of the patient in the reference group. And you also have the two quartiles, the first and the third quartile, that show you where most of the trajectories are. And so you can see that, obviously, and I haven't really showed that, but essentially here we're showing relapses and treatments as a bar. And again, if you have the infrastructure to do that, it's really it's opening lots of possibilities. And that's what I wanted to show here, is what we're doing here, is that we can actually show how one patient is progressing and make actually some sort of predictions. How we do that? It's actually very simple. We say, hey, let's look at all the patients that were like this one patient 2 years ago, or 1 year ago, and let's use that to actually make a projection about what are the most likely scenarios for that particular patient in 1 year. And you can actually refine this computation depending on the kind of drugs scenario you want to try to test. So you can do that for drug A, drug B, drug C, and actually try to use this information to mine the data in a personalized way to better make a decision about the treatment that you want to propose to the patient. It's also better to discuss what's the actual therapeutic options and their pros and their cons with the patient. So this is typically an example of our screen that enable refining the reference population. So you can, if you want to compare a female patient to only female patient, that's something that is absolutely doable. You just have to switch off the male, and we're going to resend a request to the central database to say, hey, great, we have to use less patients, but you can recompute the normality assessment, and this is possible for many other type of parameters, including disease duration, disease course, age of onset, and etc. We show you the 3D serial magnetic resonance brain imaging, so we can show in 3D the actual MRI images, and in this prototype we show T1, T2 proton density and gadolinium contrasted images. We can actually overlay some information. We show here a map of the myelin water fraction on the top of the images. So it's really about making the data available in 3D and eventually in 4D dimension when we have several exams at a time, like you have 6 in here. So this is pretty much what I wanted to tell you about this prototype and where we are. What we've been doing is we've been working in cycles to get a prototype. That's what we have for now. I usually call that like a concept car. It was a quick development. We've been working on that for the past 18 months. And we conducted two sets of pilot study, one with patient and one with clinician to try to better understand what we're doing and where we should take it. And we will start doing a second pilot to better evaluate how these kind of tools will enhance practice and change practice in the clinic. So there was a couple of hypotheses supporting these this type of pilot study. And typically about the patients, we thought that this kind of application helped patients to better understand the disease, share the data. They want to share the data for research, and they also want to compare the results to other patients. That was not granted, so we cannot ask them. And we did the same with physicians. We also thought that would really enable data-driven presentation of disease features for decision making, because the data is all here at hand, because we provide tailored comparisons that are actionable, and eventually we give access to data that are typically not so easy to get, like something that we compute from brain MRI is the annual change in brain atrophy. So we're quantifying what is usually a qualitative assessment of the brain atrophy as seen by the neuroradiologist on the MRI images. So there's a couple of questions we asked the patients, but what I wanted to do is actually before before showing to you the results that we got from patients, I thought this webcast tool is actually great, and I wanted to propose to you to answer the same questions. So if you don't mind, let's do it. And the first question I wanted to ask you is, would you feel comfortable obtaining your personal research data, even though it has not been approved for diagnostic purposes? Because many times, that's the question that was raised actually by the IRB. Even this project was funded at the federal level. Say, hey, you can't do that. This is research data. And most of the time, the consent actually explicitly say, we are not, you won't get an individual return on your data. We took kind of the alternate or the opposite assumption, saying, hey, data is just data, and if patients want to access the data, yeah, maybe we should do it. And so we asked actually the patients, and we say, hey, would you actually be okay in looking at your data, even if these data, like genetics, has no impact on clinical information, on clinical data? It's not CLIA certified, it's not approved for this kind of application. So this first question was really about accessing research data. And that's because what we believe is that this kind of technology like the bioscreen is actually blurring the frontier between what is FDA approved in a way and what is not FDA approved. I'll let you answer for a few more seconds. I have about half of you already. So the question again is, would you feel comfortable obtaining your personal research data, even though it has not been approved for diagnostic purposes? Thank you very much. A few more minutes. Raul, thank you for your question. You were asking about a couple of more answers, going very quickly. Thank you very much to all. It's actually look very similar to what we have for the patient. I'm going to close it. Okay, I won't copy that. I'll show you the result then, and I'll tell you what patients are thinking about. But essentially, we are about 90% of yes. Okay, thank you very much. So the next question we asked the patients was actually to test whether the fact that we're using a software application was actually a problem for patients. So we asked them, would you be interested in using a secure software application, a website, or a tablet application like the one we developed for the MS Bioscreen, in order to access your research data and also eventually your clinical data? Thank you very much for answering the question, and it's remarkable. We have a quarter of all of the attendance right now. It's remarkable how close you are from what our patients are saying. It's really, really interesting. I'm going to leave you another couple of minutes because there's many other questions that we asked to the patients. I had a question from Raul about patterns in healthy controls, it's actually right that we find, I suppose you're talking about genetic risk. We do find a lot of healthy controls that have very, very similar risk pattern than the patients. Unfortunately, it's only genetics. We don't have MRI information on healthy control that would enable us to find patterns, similarities, or many other metrics that we do have for patients that we could use to find similarities between controls and patients. There's some old literature saying that they found within families they found patterns, both of genetics and other features between the siblings of affected patients. So we asked them about the software. We also asked the patients okay, it's only two of the questions that I need 2 questions out of 10. So it doesn't matter, it seems that I don't have, I'm not able to push all the questions. But essentially we asked the patient about how much they wanted to have a comparison with other patients. That was question 3. How much they would be willing to enter or provide their own information, question 4, and if eventually they would like to receive data information remotely. And the other sets of questions were kind of a second generation of questions, but also question in a way. Trying to better understand why this is a personalized application. How so we asked them if there's more information or if there's area that they don't see in the data, and in particular we're developing new features and new screens about quality of life data. That's what we had in mind. It's assumed that patients will take a more active part in the decision-making process, but we want to query. That was question 2. And question 3, and maybe if we have a little bit more time at the end, I'll ask you to answer that questions. Really also question number 4 was about therapeutic strategy, right there. And question number 5 was about the fact that a lot of the information is actually use algorithm and computerized information. So it's pretty much the man versus machine, or some automated algorithm that are getting an increasing role into this data. So just let me show some of the results we got from the patients. It's remarkable that 96% of the patients actually feel very comfortable having access to their research data. About 90% feel also are interested by accessing the information using an app or a website. 90 to 93%, it's probably a little bit less in the most recent update, of the patients actually want a comparison. And we were actually surprised by how high this number is. We were expecting a little less than that. And what we actually also saw is that that's in the ones that don't want to see their comparison, it seems, and again, we're talking about 40 people here, it seems that the people that don't want to see a comparison, half of them are actually patients who already know they're not doing so well. So they don't want to see themselves in the portion of patients that are not doing so well. But there's another, about half of patients who just don't care about all this data. What they care about is the trust they have with their physician and say, great doctor, you're using this tool, it's fantastic. I'm sure you're making the decision, I'm sure you're making better decision. I feel fortunate that you're using these kind of tools, which are probably the best ones, and etc., etc. So it's really an interesting concept to see that the use of technology in a way reinforce the trust between the physician and the clinician. Lots of patients, but these are from a research cohort, are willing to enter their own information. We could think about sending their MRIs. We could think about providing information about their quality of life. And 9 out of 10 would also agree to receive information remotely and advice from the data. So we also asked the clinician about to better understand what they thought is interesting in this application. Some of them have been using the application to show to patients, and essentially what it shows is it's certainly there's certainly a great convenience factor. It's very convenient to be able to access all this information in one place. It certainly helps to empower physician authority at the point of care, in particular when they have access to data that is not typically available in regular clinics. It also has maybe some potential predictive value, but this is probably not there yet. And again, this is just a prototype we've been working on for 18 months. But what is also interesting is that they said that it's starting to shift their traditional clinical reasoning from using average results, which are the ones that are published, to to find really an individual normality assessment that is data-driven. In the interest of time, I'll go switch one or two slides. Just want to insist on the fact that we are no descriptive, maybe comparative to other patients, but we're not really predictive and prescriptive so far. But that's the start of the application. That's what we're working on now. I'll skip this one, but we're working very hard to integrate more data from clinical trial and from electronic medical records databases. So we kind of skip these two challenges because we had our own cohort and database system. So what we also have in here is really challenges to make the Bioscreen working on different kind of platforms, including different kind of EMRs, like a solution that would be plugged on the top of database using different kind of data. And so the Bioscreen has really the power to use the clinical research data and make them actually more usable than they typically are, and promote standards of integration. So again, we think that application like the MS Bioscreen for complex chronic disease really try to address the challenge of the dynamic management of the disease. It's probably applicable to other chronic conditions. And in our development, there were 3 key evolutionary phases: to visualize the data, to contextualize it, to understand the data using comparison, and to be able to interact with the interpretation and the way we do the normality assessment of the patient. I wanted to thank again all the people, more than 50 people that have been working with us and over the past 18 months. And I'll be happy to answer more questions by email, and if you want to check our website and videos online. Thank you very much for your attention. I'll take more questions. I have a couple of questions. Sorry, I'll probably select just a few of them. I'll be very happy to follow up by email. I have a question about security, which is interesting, and so the question is, we should have asked if the patient believes that the application is really secure. Yeah, and you're referring to Target that lost or was hacked for credit card information. That's very true. I think the problem is dual here. There's the security about is somebody will have access to information, which is different from the fact that many types of different information are aggregated into the same place: genetics, MRIs, and etc., etc. So these are two different questions that we don't know really how to separate, because it's not always very clear. So what patients have in mind. Sometimes they think about their credit card information because a hacker would take money from their account. It's different from being anxious about having so much information into the same place. After all, your credit card information is not just only one account, one bank, and but it's not all the history of your data, and not your past and your future and your present information. So if people have very important questions, I'll be happy to answer that or elaborate more on one aspect or the other of the bioscreen. So I also have a question about contextualization. Okay, I can close now. I'll be happy to follow up on questions about algorithms, MRI visualization technologies, and application to other disease is also very important questions. Yes, certainly it has the potential to be applied to many other diseases, and we'll be happy to one of our next steps is to try to deploy that into different centers, not only at UCSF, to be able to aggregate more data and strengthen the power of the algorithm to be ultimately predictive. So thank you very much for your attention. It's a little bit frustrating not to see the audience, but I hope we'll be in touch later on by email. Thank you very much all for your attention, and I hope we'll have opportunities to meet in meetings. It's 7:05 p.m. here in Nantes, west of France. I'll be back to the US very soon. Thank you very much.